Hey guys, it's Miss Adelini. This is a video tutorial on dihybrid crosses, and I'm sorry I am in my living room, so you might hear Andrew making dinner in the background, so hopefully that doesn't distract you. But let's go ahead and get started. You should be in your genetics notes packet, starting off where we left off with the blood type notes. So up until this point, we've been looking at monohybrid crosses, mono meaning one, so we were tracking the inheritance of just one trait at a time. Now we're going to be creating a dihybrid cross, di meaning two, and we're going to be showing the inheritance of two traits at a time. We're going to use the example of pod color and plant height, like in Mendel's pea plants. We'll say that the tall allele is dominant over the short allele, so we'll use big H for tall and little h for short. We'll say that the green allele is dominant over the yellow allele, so we'll use big G for green and little g for yellow. So for example, for our first cross, we're going to have our first parent be tall and green, and it'll be homozygous dominant for each trait. Our second parent will be short and yellow, so homozygous recessive for each trait. Probably the hardest part of doing a dihybrid cross is figuring out what needs to go on the top and the side of the box. So let's say the male gametes the sperm on the top and the female gametes the eggs on the left side. So the way that we figure out what our gametes are from our parent genotypes is we use a method that you may have used in math class called the FOIL method. Now, if you're using it in math class, you might be using it to combine two terms that are in parentheses. So you would do the first terms, then the outer terms, then the inner terms, and then the last terms. And that would help you to combine them. Now for our parent genotypes, we use FOIL by taking the first H and the first G. That's going to be our first gamete, and that's the F in FOIL. Then we take our outer H and our outer G for our next gamete. That's the O in FOIL. Then we take the inner H and the inner G for our next gamete. That's our I in FOIL. And then we take our last H and our last G, that's the L in FOIL. If we did the same thing, first H, first G, outer H, outer G, inner H, inner G, and last H, last G for our parent 2, we'd end up with these possible gametes. Notice that in the gametes, we only have one copy of each gene. We only have one of each letter. That's very important. When people are setting up a dihybrid cross, that's where they tend to get screwed up. They will do two H's and two G's. That cannot happen. Each gamete carries one copy of each gene. So let's put parent 1's gametes along the top and parent 2's gametes along the left-hand side. And when we fill in our Punnett square, for this particular cross, I'm always going to put my H's first and my G's second, and I'm always going to put any capital letter before a lowercase letter. So it just so happens that in this cross, all my gametes on top are the same, all my gametes on the left side are the same, so all of my offspring will have the same genotype. So you don't really have to fill in all the boxes. You could simply write all big H, big G, little g. And that's fine with me. So that would give you a genotype frequency of 100% big H, big G, little g, and a phenotype frequency of 100% tall, since big H stands for tall and that's dominant, and green, since big G stands for green and that's dominant. Moving on to our second example, it's a little bit more complicated. We're going to use two parents that are heterozygous for both traits. So we have to be careful when figuring out our possible gametes. I use FOIL. I take my first H and my first G, so that's big H, big G. I take my outer H and my outer G, so that's big H, little g. I take my inner H and my inner G, so that's little h, big G. And I take my last H and my last G, so that's little h, little g. I do the same thing for my second parent, who has the same genotype, so we'll end up with the same gametes. And if I put those gametes from parent 1 on the top and parent 2 on the left-hand side, I can then start filling in my offspring, remembering that I'm always putting H's first, and I'm putting capital letters before lowercase letters. All right, I would suggest pausing the video at this point and trying to fill in the offspring on your own. Then, when you're ready, I would check to make sure that your offspring match with the offspring that I have on this screen. Now that you're done pausing and you've checked your offspring with my offspring, we are going to count the number of offspring that have each phenotype, each physical trait. It would be really complicated to figure out the genotype frequencies, the combinations of letters, so I want you to cross out where it says genotype frequencies for this example. We're just going to stick to the actual physical traits. So, I am going to use a color coding system. I'm going to fill in my offspring, and if I was doing this on paper, I might have a highlighter. I am going to color code all my individuals that are going to be tall and green, so at least one big H, at least one big G, in the color blue. Then I'm going to fill in all of my offspring that are tall and yellow, a big H, little g, in yellow. All my offspring that are short and green, little h, big G, in orange or peach. And then my final offspring is short and yellow, little h, little g. Again, I would probably pause it at this point to make sure that you are clear on what I have done with the color coding system. Another way to do this would be to count the number of a particular phenotype. So you might say, hey, I am going to go count all of the tall and green offspring. And you would cross it out as you go. So I'd count this one, 1, cross out, 2, cross out, 3, cross out. And once you have the number for that phenotype, you can start counting the next one. But making sure not to double count any offspring by crossing them out. So either way, no matter what method I use, I end up with a 9-3-3-1 ratio. 9 out of my 16 offspring are tall and green, so they have both dominant traits. 3 out of my offspring are tall and yellow, so they are dominant recessive. 3 out of my offspring are short and green, so they are recessive dominant. And then 1 out of my offspring is short and yellow, so recessive recessive. The interesting thing is when Mendel did his pea plant crosses, he found that when he crossed 2 parents who were heterozygous for 2 traits, he always ended up with this 9-3-3-1 ratio. 9 dominant dominant, 3 dominant recessive, 3 recessive dominant, and 1 recessive recessive. Crazy. Okay, so this is where we're gonna stop with the actual notes. And we're going to go to the worksheet, the Dihybrid worksheet. And I've created a key, and I'm going to go through the key for the first side with you and for the first problem on the back side. Okay, I'm back. I had to ask Andrew if dinner was ready. All right, so let's go ahead and get started. So at the top, they tell us what traits we're looking at. The trait of height, tall being dominant over short, and seed color, this time yellow is going to be dominant, big Y, and green is going to be recessive, little y. They also tell us the parent plants that we're going to be using. Our first parent is heterozygous tall, so big T, little t, and homozygous for yellow seeds, big Y, big Y. Our second parent is short, little t, and heterozygous for yellow seeds, big Y, little y. Number 2 is really easy. They already told us our phenotypes of the parents up here in the information section. My first plant is tall and yellow. My second one is short and yellow. So before I set up my dihybrid Punnett square, I need to take my parent genotypes and foil them to find my gametes. That's why I wrote use foil on the side. So this is my parent genotype for plant 1. So I'll take my first T and first Y, big T, big Y. My outer T, outer Y, big T, big Y. My inner T, inner Y, little t, big Y. And my last T and last Y, little t, big Y. And that gives me these 4 gametes listed under number 3. Then I'm going to take my genotype for plant 2, little t, big Y, little y. I'm going to FOIL that. My first T, my first Y gives me little t, big Y. My outer T, outer Y gives me little t, little y. My inner T, inner Y gives me little t, big Y. And my last T and last Y gives me little t, little y. So that's going to give me my 4 gametes that I see under number 4. The next thing that I need to do is I will put my gametes from parent 1 up across the top of my dihybrid Punnett square. Then I'll put my gametes for parent 2 along the left side of my Punnett square. I would pause the video right now and I would fill in your offspring genotypes. And then I would check to make sure that your offspring genotypes match with mine. Remember to always put your T's first in this case and your Y's second, and to put capital letters before lowercase letters. Okay, welcome back. Hopefully your offspring genotypes matched mine. Instead of using a color coding system, when I was counting, I simply crossed off each box as I went. So I've actually asked you to figure out the genotype ratio for this one. So I counted my numbers of each genotype, and when I did that, I ended up with the following genotype ratios. I found out that 4 out of my 16 babies, or 1/4, were big T, big Y, little y. 4 out of my 16, or 1 out of 4, were big T, big Y, little y. A quarter, 4 out of 16, were little t, big Y, big Y. And a quarter, or 4 out of 16, were little t, big Y, little y. I don't have to go through and count again to determine my phenotype ratios. I can just use my genotype ratios. So my first 2 genotypes shown right here would give me tall yellow plants. So I simply add 1/4 to 1/4, and I realize that half of my offspring will be tall and yellow. My second 2 genotypes would both give me short and yellow plants. So again, I'll add 1/4 to 1/4, and that gives me half of my offspring being short and yellow. Okay. I again would like to try the first problem on the back of the page, and then I'm going to have you do the last 2 problems on your own. So we're going to be looking at 2 traits in mice, the running trait, big R, and the waltzing trait, little r, where they run around in circles. Hair or fur color is our other trait. We'll use black hair, big B, to be dominant over brown hair, little b. So we're going to be crossing in our first cross a heterozygous running, heterozygous black mouse. So this is my first parent right here under parent genotypes. Heterozygous running, heterozygous black would give me a genotype of big R, big B, little b. My second parent is homozygous running, homozygous black, so that gives me a genotype right here of big R, big B, big B. If I use the FOIL method for my first parent, I end up with the following gametes along the top. My first R, first B is big R, big B. my outer R, outer B, big R, little b. My inner R, inner B, little r, big B. And my last R, last B, little r, little b. If I use the FOIL method for my second parent, I end up with 4 of the same gamete, big R, big B. And now would be a good time to pause, take a minute to fill in your offspring genotypes, and check your answers once you've done so. Okay, now that you've checked your answers, we're going to fill in the phenotype ratio. If you count, you'll notice that all of the offspring have at least 1 big R and at least 1 big B. That means all of the offspring show both dominant traits, running and black. So our phenotype ratio is 100% running and black. All right, that concludes our video tutorial on dihybrid crosses, and I hope this was helpful. All right, talk to you guys later. Bye-bye.